In part 2 of Understanding Inheritance, we're going to get into the modeling. So how are the traits modeled? And then we're going to end up with talking about variations in Mendel's theories, which means things that were discovered not by Mendel, but because of his groundwork, we were able to discover some other things in genetics that weren't just dominant and recessive. That's one of the main things. They weren't dominant recessive. So let's first talk about modeling. In modeling inheritance, we usually use one of two things, and one thing we use is a Punnett square. The Punnett square is a model used to predict possible genotypes and phenotypes of offspring. And we're going to learn how to do this with these models in class. So I'm going to teach those how-tos in class. But here's an example. A Punnett square, the ones that we are going to do, will have 4 squares. But there can be 16 squares and more. So you can have Punnett squares as large as you need it for the type of genetic problem that you are doing. Notice we're using letters just like we talked about in the last vodcast. The capital letter, remember what that stands for? Dominant allele. And the lowercase letter? Recessive allele. The other modeling that we have is a pedigree chart. And a pedigree chart is going to show the phenotypes of genetically related family members. And they use shading and non-shading to show the phenotypes. All right, here's the variations. These are some things that Mendel did not learn and did not recognize when he was working on the pea plants. And we're going to talk about each of these and what they mean. Some of them we will actually be doing some genetic problems with them. There's incomplete dominance, there's codominance, there's polygenic inheritance, and there's sex-linked traits. Incomplete dominance. Okay, sometimes traits appear to be blends. See, with Mendel it wasn't. Whenever he got crossed 2 purebreds, crossed 2 homozygous parents, he always got the dominant trait that showed up. But later on, when people started crossing some different flowers, such as 4 o'clock flowers, and they crossed a red with a white, you would expect, if Mendel's theories were the only theories that were possible, that one of those colors would be dominant and would show up. But instead they got pink. So that's called incomplete dominance. The alleles show incomplete dominance when the offspring's phenotype is a blend of the parent's phenotype. Now, codominance. What do you think codominance would mean? It occurs when both of the alleles can be observed in a phenotype. This is going to be easier seen when I show you the pictures, but that usually means it's going to be splotchy. In other words, you're not going to have just one color dominating. You're not going to have them blending and mixing to a different phenotype, but you're going to have splotches of each phenotype showing up in the offspring. Okay, incomplete dominance. It's a type of inheritance in which the genes expressing a particular characteristic are neither dominant nor recessive. That should really be the alleles expressing. 2 traits combine, they blend together. So the key thing about incomplete dominance is a blending of the 2 traits. It's when offspring of 2 homozygous parents show an intermediate phenotype. So it's going to be a phenotype that neither of those parents had. It's going to be a 3rd phenotype showing up. Here's a good example. 4 o'clock flowers, snapdragons are examples of plants that when you take and mix, and it doesn't always mix according to our color scheme. Okay? So God's color scheme is not always what the alleles are going to produce. You know, a lot of times, yes, red and white produces pink, but you're going to have a red radish and a white radish that actually produces purple radishes. So it's not a pink radish. See the codominance here? In codominance, the dominant and the recessive traits are both expressed because there's not really a recessive trait. Both traits are showing up. One is not dominating over the other. So obviously, if you look at these cattle, one of the parents was a brownish red color and the other parent was white. And that gave these what are called roan cattle, which are heterozygous. Here's in the flowers. In incomplete dominance, neither allele is fully dominant. This is different from codominance, in which both alleles are fully expressed, resulting in organisms that display the characteristics of both parents. So if you look on the left there, you've got a red carnation crossed with a white carnation on the right, and in the middle is the blend. So you got the homozygous red, the homozygous white, producing the heterozygous offspring of pink. So which is that? That's incomplete dominance. I'm not sure what type of flower is over here in the lower right, but those parents were red and white as well. But notice how those parents' alleles did not blend in this type of flower. They actually both are expressing. You see splotches of the reddish or pinkish, I guess it's a reddish, and then you have splotches of the white. And so how do we know which is codominance, which is, incomplete dominance? It's just by doing crosses and watching and seeing what alleles produces what in the offspring. And of course it was all set up by God at creation. In codominance, both alleles are expressed independently, and they are uniquely recognizable, which you see not on the three pictures on the left, but with the one picture of there with the splotches of red and white. So codominance, another example is blood type. Human blood type is, the ABO grouping, is determined by more than one allele. And we probably will not get into these types of problems, but you can see how they write the genotypes and what the phenotype is. If you have type A blood, you can be homozygous or you can be heterozygous for type A. If you have type B, you're going to be either homozygous or heterozygous. If you're type O, there's only one example, and that is a heterozygous, a purebred recessive. And then you have the codominance showing up when you have the type AB, because both the A factor and the B factor, the alleles, are going to be expressed in that blood type. Now polygenic. Poly means many. Polygenic, think of polygenes, many genes, inheritance. Not everything is as simple as what Mendel dealt with. All the 7 traits that he picked with his pea plants, they had only 2 alleles for a gene. That was it. So there was one or the other, no other possibilities. But when they started looking at other organisms and other traits, they found out it's not that simple. There are a lot of traits that are actually more than one gene. So it's 2 or more genes, which means there would be 4 alleles, 6 alleles, and so forth. And you need to know the examples that are in humans. But first let's go over the definition. Polygenic inheritance, 2 or more genes producing a single trait. It occurs when multiple genes determine the phenotype. And know the examples in humans. This is just a few. Obviously we don't have time to discuss every single one, but these are probably major ones that you would think of. Hair color. One book that I read said it could be as many as 12 genes producing our hair color. Skin color, eye color, our height, our intelligence, our IQ, our body build, which is the basic size of your bones. So all of those are the result of more than one gene. It's 2 or more. We will not be working these types of crosses. And when you get to biology, you will work dihybrid crosses, which will deal with 2 genes instead of just 1. All the problems we're going to do is just 1 gene, 2 alleles. Another thing that can affect genes and how they are expressed is the environment. An organism's environment can affect its phenotype. Now, this is not with all genes, but there are certain ones. Some examples of environmental factors that affect phenotype are soil type that a flower is growing in, or the time of year that a butterfly develops. So those are 2 examples. I'm going to use the example of the plant because I have a hydrangea at home, so I know how it works. With the hydrangeas, when you read this, you don't need to write this down or anything. It is the pH of the soil that is actually going to determine what flower color the hydrangea is going to be. Now, since I have a real high pH acidic soil, mine are a deep, actually not even blue, they turn deep purple a lot of times. But sometimes they might be blue. And you can even have a mixture in the same soil. So as it says, in strongly acidic soil, the flowers turn blue, the pH is below 6. In alkaline soil, where the pH is above 7, the flowers turn pink or even red. And in strongly acidic, and that's what I have, soil or neutral soil, the blooms may be purple or a mix of blue and pink on a single shrub. And I get the deep purple because I have a lot of pine trees, and the pine needles are what actually make the soil more acidic.